First of all, I would like to thank all the organizers for the invitation and the audience for sharing their time. My name is Mehran Moghadam. I'm the CEO and founder of OROX BioSciences. And this afternoon, I would like to share with you our efforts around discovery of novel bifunctional small molecules. These are dual inhibitors as a first-in-class opportunity in cancer and beyond.
Can I interrupt you?
Sure.
Can you explain what that is for people who are in the audience? Because we have a wide variety of different kinds of people.
Of course. So in the world of pharmaceutical industry, we kind of divide the drugs into small molecules or large or biologics. Small molecules are generally made by the chemists versus the biologics that are made in vats by organisms and biologists. So the way we are designing our molecules purposefully is to inhibit two targets, two therapeutic targets at a time. Therefore, you're interfering with disease process at two different nodes, and you have synergy because of that. And these have traditionally been difficult to design, and there's novelty here because of what I'm about to explain. Is that okay?
Yeah, that's perfect. Thank you so much. That's awesome.
So, as a summary, just in case you guys are going to tune me out after this slide, we have a highly experienced team with many years in the pharma industry. We have a preclinical development candidate for bladder cancer and preclinical pharmacology data on multiple animal models in cancer. We have IP-protected drug discovery platform technology, which has enabled us to make a collection of these first-in-class small molecules. And we feel pretty good about the risk, the clinical safety risk of our molecules. And I explain why. But before I leave this slide, I wanted to also introduce another message here, which I'm going to build on soon. And that is that our lead molecule, in addition to its ability by itself to inhibit tumor in animal models, when used in combination with checkpoint inhibitors, these are biologics, as well as the general chemotherapeutic agents like cisplatin, they can increase efficacy and reduce toxicity of those agents. And I'll come back to this message later. So, what problem we are solving? As you all know, there are many areas of unmet medical need in cancer still. Also, refractory, recurrent, and metastatic cancers are a true challenge. They have not been adequately addressed. And efficacy and toxicity of cancer therapy go hand in hand. Therefore, you don't have a nice therapeutic index or safety window. One point that is interesting to us is that the cancer therapeutics generally, I refer to them as sledgehammers. You hit the tumor with them and you kill the tumor, but you create a lot of tumor cell debris. And these cell debris then stimulate the immune system to put out what is referred to as cytokines. And these then stimulate primary and metastatic tumor growth again. So it goes in full circle and defeats the whole purpose. This phenomenon was explained the first time in 1956, still not adequately addressed. We believe that our lead molecule and our strategy addresses most, if not all, of these concerns. So now I'm getting into a bit of biochemistry here. The point of this whole slide is that we have two targets that we are going after. One is referred to as SEH, soluble epoxy hydrolase. And the other one is COX-2, cyclooxygenase enzyme. Both of these enzymes are well known, and it's known that when you inhibit these, you promote a broad range of beneficial responses pharmacologically. In terms of safety, clinical or preclinical data around SEH shows that there are no major concerns when it comes to safety. And also in COX-2 inhibition, this target came under microscope in 2004 when Merck voluntarily withdrew Vioxx, rifacoxib, due to cardiovascular risk concerns. However, in 2005, FDA concluded that, and this was a white paper, that COX-2 selective inhibition by drugs such as Vioxx or Celebrex are similar to the risks that you observe in marketed NSAIDs such as aspirin, and which is why we still have 6 marketed drugs in this area, including Celebrex. And in FDA, in 2015, reiterated that view. So we are not concerned about the safety in the clinic. I'm going to take a shortcut now and come to some recent data that we have generated and point out that in numerous preclinical tumor models, including ovarian, hepatocellular, bladder, lung, and pancreatic cancer, our lead molecule OX1 has been able to mitigate angiogenic and inflammatory cytokines. These are needed for tumors to grow. Also, they have stimulated macrophage phagocytosis, or these are the immune cells that eat the cell debris or even cancer cells. You're able to stimulate their activity and suppress endoplasmic reticulum stress, which is a factor in developing cancer. All in all, using this molecule, we've been able to modify the tumor microenvironment in favor of the host. All of that without major body loss, which you observe with other agents. And then this was the point that I brought up before: when co-administered with checkpoint inhibitors or chemotherapy agents, OX1 can significantly enhance both efficacy and safety. Here's some data just to substantiate some of these claims that I just made. In syngeneic orthotopic mouse model of bladder cancer, and this was resistant bladder cancer, we observed that when we use a checkpoint inhibitor, these are drugs that are on the market, there was no activity. However, OX1 and chemo agents had moderate activity in controlling tumor volume, but when you combine OX1, PD-1, and chemo, you reduce tumor volume in these experiments. So the effect was in tumor volume and tumor weight. You could hardly weigh the tumors at the end of these experiments. The same thing happened when you use another checkpoint inhibitor, anti-CTLA-4. So I won't go over that anymore. As far as safety, if you took male and female mice and dosed them with chemotherapy agents, shown in red here, they would lose a significant amount of weight in a short period of time, which indicates toxicity of this agent to these animals. But if you dosed that same chemotherapy agent, for example, cisplatin, with OX1, our lead molecule, there was a protective effect, and these animals did not lose any body weight. This protective effect was also shown for liver protection, in that when we measured ALT, which is an enzyme inside liver, which gets released into the bloodstream when there is toxicity, chemotherapy agents can rise the levels of these enzymes, but if you dose the same agent with OX1, there was liver protection. So that did not happen. These are very important features for us. So as far as our programs for bladder cancer, we have OX1 as a nominated drug, and the mode of application would be intravesical, meaning, and this is normal for bladder cancer, it's actually administered into the bladder itself. What we need is to raise money at this point to go through IND enabling studies, file IND, and then finish our phase 1 clinical trial. And so our need is a total of $40 million in tranches, however. We have other programs, ovarian, hepatocellular, and lung cancer, which are not as advanced, and these are not as labor intense, as capital intensive to develop them to where the bladder cancer is. So we do have a need for developing these as well. For bladder cancer, in 2026, the projected market for only North America in early stage bladder cancer is $1.2 billion per year, but this market can be expanded by moving to global market as well as treating the more advanced cancer. How we differentiate ourselves is that our primary target, SEH, is a novel target for oncology. We are the only company that's using this target for oncology. And in fact, we are the only company that uses the bifunctional SEH inhibitors, the dual inhibitors. And we do have a collection of these inhibitors in our chemical library. In terms of intellectual property, we have licensed 2 patents from University of California, and we have developed our own IP, and we are still in the process of continuing that. Our team is highly experienced. I myself have been in the industry for about 27 years. My partner, Greg Reyes, has over 35 years of experience in drug discovery and development. Neil Raheja is our medicinal chemist with over 23 years of experience in the industry. Our academic partner is Bruce Hammack. He is the discoverer of our primary target, as well as a member of National Academy of Science. And we have key advisors in several reputable organizations, such as School of Medicine at UC Davis, Beth Israel at Harvard, and Cedars-Sinai. This is our ask slide. As I mentioned, our need to finish the preclinical studies that are listed here and go through phase 1 is a total of 14 million. However, we are doing this in tranches. And with that, I would like to thank you and entertain any questions that you might have.